to another lecture of the course Aquaculture. Today I shall be talking about the culture method part 2 of farming of pearl oyster, Pinctada fucata. We have earlier discussed about the biology of Pinctada fucata, the pearl oyster, as well as we have discussed about the culture method of pearl oyster, Pinctada fucata, its hatchery operation and other things. Now we are focusing more towards pearl formation, the purpose for which we are doing all this pearl oyster farming and related things. The pearl formation is the main part what we are going to discuss today. In fact, what happens that the pearl formation in case of these oysters that occurs as a sort of natural activity when any foreign material enters into the body, like parasites, sand grains, broken twigs of sea seeds or whatever seaweeds, and small insects enter the body of an oyster and adhere to a part of its mantle. If any foreign body somehow enters into the body of the oyster and adheres to part of its mantle, then what happens? The mantle, as a natural response, the mantle epithelium at once encloses it, the foreign body, like a sac. So there is a sac formation around that foreign body as soon as it attaches to the mantle. It enters into the body and attaches to the mantle. The mantle epithelium starts to secrete concentric layers of nacre around it. This is the main concept of how pearl formation occurs. If any foreign body has entered into the body of pearl oyster and it adheres to the mantle, what happens? The mantle epithelium secretes concentric layers of nacre around it and it encloses the foreign body like a sac. It is carried out from the defense point of view, in fact, and in fact, we are what we are taking as a pearl. That entire process was initiated by the pearl oyster as a defense mechanism for some foreign body which has entered into its body. This is a beautiful picture that you must have seen many times at many places in many of the literature, published literature, that when a foreign body enters, then the mantle epithelium that starts enclosing it, and the layers of nacre is deposited around that foreign body. That is gradual and process that ultimately this foreign body, along with the nacre which has been secreted around that foreign body, that becomes our pearl. And that is why sometimes you must have heard that the more time a foreign body remains within the oyster, it becomes a better pearl. There will be more and more layers of nacre around it, and the pearl will be of the desired size that you require, or sometimes that can alter, that can be of the desired shape also, what you wanted. Collection and rearing of oysters, that is in the artificial insertion of nucleus, that if we have to do it, how to do it? Then first we will have to collect and rear the oysters. Hatchery source, wild or hatchery reared, any oyster you can take. It depends on, I discussed it in detail in my previous lecture, that how we can collect the oysters which are suitable for artificial insertion of the nucleus, which can be used for pearl formation. Oyster stage, immediate post-spawning recovery phase is considered better immediately after spawning. when they are in the recovery phase, or early phase of gametogenesis. This should be taken into account that if these kind of conditions are given, then we should follow the steps and collect only those oysters which have, spawned lately, and which are now in the recovery phase, or they are in the early phases of gametogenesis. Then the size, accordingly the size will be 20 to 25 grams. Oysters are cleaned and then reared for 10 to 20 days. Generally, special rearing cages are preferred for special. That means you will have to take care of them separately, and cages are well protected from natural enemies. If you are doing it in the natural conditions, if you are putting the cages in the natural conditions, then of course there are many wild aquatic animals that may damage, that may feed upon these oysters. So you will have to take care accordingly that what kind of these aquatic animals you are having in that area, and that we need to protect them. This can be done by covering them with the netting of cotton. So natural enemies like predators like octopus, seal, or other fishes like devilfish, they may not attack them. Fitness of oyster for operation. Selected oysters should be healthy. That is the first point. Strong enough to overcome shock. When we are handling any aquatic animal for any purpose, and here we have to perform surgery also, so they should be hardy enough, they should be strong, so that they can overcome the stress which is being put upon them. That is one of the very important things. We should never in fact select those oysters which are looking very weak, or which are appearing diseased, which are not very healthy, which are not showing the appropriate weight according to their age. So all these care should be taken. Ovary and testes should be rid of, dipped into cold and warm water, very important, alternately to eject their gametes. This process is also very important. Before an operation, oysters are kept under the stress of suffocation to open their shell. They naturally do not open their shell when they feel threatened. So if you are putting them in a little bit of suffocation, in fact you have to manipulate the conditions little bit in order to get what you are desiring. So in order to open the shell, if shell is not open, you cannot put the nucleus. So a little bit of stress you can apply, but that should not be enough for weakening the shell, or putting the stress too much that the oyster does not respond as you wanted. When the shell opens, a bamboo peg is inserted. When the shell is open, we simply put something in between the shell, bamboo peg, of appropriate size. That size should be taken into account, so that it is not, you are not opening the shell too much, that may damage the muscles. And if the shell gets injured, then your operation may not be that successful. Preparation of graft tissue. How we prepare the graft tissue? A piece of tissue inserted inside the mantle. A strip of about 7 by 0.75 centimeter is cut from the edge of the mantle of the donor oyster by a sharp knife. First we cut 7 by 0.75 centimeter strip, and from the edge of the mantle of donor oyster. This is cleaned and washed to remove adhering nucleus, and wiped off by sponge. You can do remove the border of the gill piece with a sharp knife, so as to make the desired size what you wanted. Preparation of the graft tissue, here it is shown. Trim the tissue now to a strip of 2 to 3 centimeter long that you want. and again cut transversely into small squares. From this 2 to 3 centimeter long, you can cut small squares of the strip according to the size of the nucleus that you wish to put into the body of the oyster. So according to the size of the nucleus that you will have to cut this the strip also accordingly. The size of the strip should be adjusted. Keep these squares in seawater at 22 degrees centigrade for 48 hours. Outer edges of the graft mantle that we have just cut down in the form of the strip that contain nacre secreting cells. That is the the graft of the mantle that we are putting it along with the nucleus into the body of the pearl oyster is basically to induce the nacre formation. It will induce the nacre formation from the recipient pearl oyster. So that helps to expedite the process. Any small particle may function as the nucleus. We have previously discussed that any foreign material, if it enters and adheres to the mantle, the process of nacre formation and covering it into a sac starts, and that is the beginning of the pearl formation. Best nucleus, calcareous nature. These are found by thick shells of other mollusks, usually freshwater mussels with heavy deposition of calcareous shells. Here you can see that the best nucleus which is considered for putting it into the body of the pearl oyster should be calcareous in nature. And we all know that calcium deposition or calcareous shells are found best in case of freshwater mussels. So usually we collect freshwater mussels. The nucleus is cut according to the size and shape that you want, and ultimately that is put inside the body of the pearl oyster. So these are easily available. In case of India, we are blessed with huge resources of any kind. Japan, for example, Japan is not having that much of these freshwater resources that can be utilized for preparing the nucleus. So it depends on other countries like USA for good quality shells, but that is necessary for getting a good quality pearl. But we are having, in fact, the resources which we can utilize for the purpose. The number of nuclei to be implanted should be decided before operation. There are, different methods, different approaches adopted by different people in different regions. So, in fact, a number of modified technologies are also there. Nowadays, you will also find some people, entrepreneurs who are running such kind of business. They are also modifying the methodology a little bit according to their own requirement of the site and the type of oyster they are having and the demand. So there may be single implantation. If we do go for single implantation, 6 to 7 millimeter nuclei. For double implantation, we have 4 to 6 millimeter nuclei, and if we are opting for multiple implantation, then 2 to 3 millimeter nuclei can be used. There are two methods of doing this, and method 1 and method 2 that we shall be discussing here. Now start with method 1. Here what we do, I can just tell you how the things are going on one by one, step by step. Fix the oyster in desk clamp with the right valve facing upward. Right valve should be facing upward and it should be fixed on a desk clamp. The thing is that the workability should be easy. The access of the technician to that portion where we are going to put the nucleus should be easy. Expose the foot and main body mass by touching the mantle folds. If foot and main body can be exposed, make an incision into the foot epithelium and slender channel into the main mass. Suddenly place the graft tissue piece into the channel and then the nucleus over it. Remove the bamboo peg. Remember what we discussed, that when the valve was open, then we put something of, say for example, bamboo peg, a small bamboo peg of the desired size, in order to keep the valve open. Now graft tissue and nucleus you have put in, so the bamboo peg is removed, so the oyster shell closes automatically. The operation period should not be done more, beyond 30 minutes or so. It should be done easily and in less time. That will be putting less stress to the fish, to the oyster. Now, second method is also there. Some people are using speculum, a tool that is often used for this purpose, into the oyster's posteroventral corner and open it by moving the gap regulator ring of the speculum backward. Avoid opening the oyster too much, since I told you in the beginning also, when we are opening the oyster, it should not be open very widely, because the adductor muscles may break, and if the muscles gets injured or break, then there will be different problem. Place the oyster with speculum on the clamp. You can place the oyster properly. To avoid slipping, put the oyster correctly between the clamp's two plates. So that can be very easily done if you are using speculum. The speculum is now toward the technician's left side, and now what is being done, that hook the needle around the tip of your foot and slowly draw it up so that the base of your foot is slightly raised. Hold the needle in place until the process is finished. So that is the process being explained, that how speculum is used. I have provided here the reference also. If you wish to go in further details, you can visit the web page. Make a sharp incision at the base of the foot with the right hand, holding the incision cum grafting needle's oval knife end. Pass the needle into this incision underneath the outer skin and slowly and carefully carve a route through the gonad connective tissue to the implantation location. Now gently extract the needle. Now pick up a piece of graft tissue from the block with the tip of the needle and gently put it through the passage made in the gonad. When the needle reaches the location, gently deflect it in order to drop the graft into the place. Now withdraw the needle from there. Lift a nucleus with the wet cup end of the appropriate nucleus implantation needle and gently put into the gonad. When you approach the site, gently deflect the needle until the nucleus drops. At this point, the nucleus must be in touch with the outer epithelium of the mantle tissue that was grafted into the gonad in the previous step, earlier what we have seen. Pull the needle carefully through the passage. Smooth the incision with the cup end, allowing the two margins to come into touch. They should touch each other. The nucleus implantation operation is now complete. Remove the oyster from the clamp now, then withdraw the speculum by sliding the gap regulator ring forward. Place the oyster in clean seawater now. Now, either of the methods, whatever methods you are using to put the nucleus, the ultimate aim is the mantle epithelium should be induced to form a sac and a nacre around it, and that will ultimately form the pearl. But after surgery is over, that means when we are doing surgery, we are taking the animal out of water and we are doing many things which are putting much stress to the animal. So you will have to take care that the animal feels comfortable after passage of the time. So post operational care to these animals is very important. Freshly operated oyster should be left alone for a few days. Don't disturb uh, the it's like they are in a kind of shock, and they should be left alone for a few days. They should be placed in plastic troughs or FRP tanks if kept in the laboratory, where seawater flows gently. Other conditions we should try to maintain that whatever desirable range of other parameters they need, water quality parameters and all, they should be getting that. Seawater has to be changed frequently if flow through system is not available, but preferably go for the flow through system. Maybe the same water you can filter and rerun again after doing everything, but try to maintain flow through. Oysters should be examined properly, and dead ones should be removed. You may get mortality if the surgical operations have not been carefully performed, and if something like this happens, then dead ones should be removed as quickly as possible. If placed in cages, 3000 to 3600 nucleated oysters should be kept in different cages and suspended in seawater 2 to 3 meters deep. The length of the culture period depends on the size of the nuclei inserted. For 2 to 7 millimeter diameter nuclei, 3 to 24 months. It depends on what was the diameter or size of the nucleus that you have inserted. Size, shape, and weight you can also consider accordingly. But weight is not that important. The size basically is important. And if you are using 2 to 7 millimeter, then 3 to 24 months are okay. Harvesting of pearls, that can be done by machines or manually. Manually collected by initially separating the two shell valves. First you will have to separate the two shell valves, then cut the adductor muscles. Earlier we were taking care that adductor muscles are not injured. Making an incision into the gonad, on the gonad, and squeeze the pearl out. By machine, how we can do it? This usually works by dissolving the oyster's soft body parts with the use of chemicals, while the pearls remain as they are and become easily extractable. When other soft tissues are dissolved, then of course the pearl will remain. In case the oysters need to be reused, but that will not work if you wish to reuse the oyster, then the pearls are carefully removed by opening the pearl sac through the gonad, taking care not to damage nor stress the oyster. You will stress the oyster, but not to that extent. And you can very skillfully, you will have to remove the pearl from the oyster and then take care of that pearl oyster for some time. If it is okay, then you can use the same oyster again. So this is a photograph where harvesting has been shown. Now the quality of the pearl. Pearls are of variable shapes and sizes, and we all know that if you go into the market looking for pearl, then you will get to see the prices ranging from very low to very high. Color white, cream, red, pink, red, a lot of colors are also now available, even black pearl that is very costly. Spherical pearls of rainbow color are rare. Linga pearls are considered by many as the best quality pearls obtained from marine oysters, and usually it is considered that freshwater pearls are not as valuable commercially as marine water pearls. There are certain ways to grade and market the pearls. If the pearls have to be marketed, then they are purchased according to the grade that has been produced. So there are different grades. The quality pearls are AAA grade, excellent luster. If you are having excellent quality, that is considered as AAA grade. They have excellent luster, no surface flaws, and good symmetry characterize the highest grade with superb attributes. The one which is considered the best is given AAA grade. Now, second, AA grade. Good quality, of course, slightly less than the AAA grade. Good luster, homogeneous coloration with a few surface defects that may be found, but not very easily. A grade, medium quality, good luster, non-uniformity in coloration with some surface imperfections and poor symmetry. In fact, when you harvest the pearls, then you will have to grade accordingly, and that is the way you will get the price for that. Then we have B grade, when good luster is there but with irregular surface and coloration, as well as a few surface flaws can also be seen. So that is given B grade. And the last one, that is NC grade, that is not suitable commercially, or you can say no commercial value, people say, but uh, that will have, you can say low commercial value or no commercial value. The shine is low, nacre layer is weak, and serious flaws on the surface. So that is graded as NC, and that is the grade which no pearl farmer will like to have. So that's all in the pearl farming or pearl oyster farming details, culture of pearl oyster, and pearl farming methods. So thank you so much.